In today's episode of Nobel Prize 2020, we'll talk about the Nobel Prize in Chemistry. On the 7th of October 2020, the Royal Swedish Academy of Sciences decided to award the Nobel Prize in Chemistry to Emmanuelle Charpentier and Jennifer Doudna for the development of a method for genome editing. Emmanuelle Charpentier and Jennifer A. Doudna have discovered one of gene technology's sharpest tools, the CRISPR-Cas9 genetic scissors. Using these, researchers can change the DNA of animals, plants and microorganisms with extremely high precision. This technology has had a revolutionary impact on the life sciences, is contributing to new cancer therapies, and may make the dream of curing inherited diseases come true. Researchers need to modify genes in cells if they are to find out about life's inner workings. This used to be time-consuming, difficult and sometimes impossible work. Using the CRISPR-Cas9 genetic scissors, it is now possible to change the code of life over the course of a few weeks. It all started when Doudna got to know about bacteria's ancient immune system from one of her colleagues. Researchers found repetitive DNA sequences that are surprisingly well preserved. The same code appeared over and over again, but between the repetitions there are unique sequences that differ. These arrays of repeated sequences are called clustered regularly interspaced short palindromic repeats, abbreviated as CRISPR. The interesting thing is that the unique, non-repetitive sequences in CRISPR appear to match the genetic code of various viruses. The hypothesis is that if a bacterium has succeeded in surviving a virus infection, it adds a piece of the virus genetic code into its genome as a memory of the infection. No one yet knows how all this works, but the suspicion is that the mechanism used by bacteria to neutralize a virus is similar to that studied by Doudna, RNA interference. What Doudna found interesting is that the genes that researchers have found in addition to the CRISPR sequences called CRISPR-associated, abbreviated as Cas, are very similar to genes that code for already known proteins that specialize in unwinding and cutting up DNA. So do the Cas proteins have the same function? Do they cleave virus DNA? She put her research group to work and, after a few years, they succeeded in revealing the function of several different Cas proteins. In parallel, a handful of other research groups at other universities were studying the newly discovered CRISPR-Cas system. Their mapping showed that bacteria's immune systems can take very different forms. The CRISPR-Cas system studied by Doudna belonged to Class 1. It is a complex machinery that requires many different Cas proteins to disarm a virus. The Class 2 systems are significantly simpler because they need fewer proteins. In another part of the world, Emmanuelle Charpentier mapped the small RNA found in S. pyogens. The results gave her a lot to think about, because one of the small RNA molecules that exists in large amounts in this bacterium is an as yet unknown variant. And the genetic code for this RNA is very close to the peculiar CRISPR sequence in the bacterium's genome. Her research group initiated microbiological detective work to map the CRISPR system in S. pyogens. This system, which belongs to Class 2, was already known to only require a single Cas protein, Cas9, to cleave virus DNA. Charpentier showed that the unknown RNA molecule, named transactivating CRISPR RNA or tracer RNA, also has a decisive function. It base pairs with the CRISPR RNA to form a functional guide RNA. Cas9 uses the tracer RNA portion of the guide as a handle, while the CRISPR RNA spacer sequence directs the complex to a matching viral sequence. In 2011, Charpentier collaborated with Doudna. Their suspicion was that CRISPR needed to identify a virus DNA and that Cas9 is the scissor that cuts up the DNA molecule. However, nothing happened when they tested this in vitro. The DNA molecule remained intact. After a great deal of brainstorming and numerous failed experiments, the researchers finally added tracer RNA to their tests. Previously, they believed that was only necessary when CRISPR RNA was cleaved into its active form, But once Cas9 had access to tracer RNA, what everyone was waiting for actually happened. The DNA molecule was cleaved into two parts. This tool has contributed to many important discoveries in basic research, and plant researchers have been able to develop crops that withstand mold, pests and drought. In medicine, clinical trials of new cancer therapies are underway, and the dream of being able to cure inherited diseases is about to come true. These genetic scissors have taken the life sciences into a new epoch and, in many ways, are bringing the greatest benefit to humankind.